For the LB amp cultures, both LB and LB amp plates grew many colonies. However, for the LB cultures, only the LB plate grew many colonies.
For comparison A, there was a significant difference between the amount of colonies in LB and LB/AMP plates when comparing LB cultures, supporting our predictions that there would be differences in the number of colonies. For comparison B, there was not a significant difference between the amount of colonies in LB and LB/AMP plates when comparing LB/AMP cultures, supporting our predictions that there would be no difference in the number of colonies. For comparison B, there is a significant difference between the amount of colonies on the LB plates when comparing LB and LB/AMP cultures, which does not support our predictions that there would be no difference in the number of colonies.
For prediction 1, there is a significant difference between the number of LB culture colonies on the two plates because the non-resistant bacteria can only live and grow in an environment without antibiotics. For prediction 2, there is not a significant difference between the numbers of LB/AMP culture colonies on both plates because resistant bacteria can survive and reproduce in environments with and without antibiotics, meaning differences in the amount of colonies is due purely to chance. For prediction 3, a significant difference in the amounts of colonies would normally suggest one culture is better able to thrive in a given environment. However, with no selective pressure against either culture on the LB plate, the differences may be due to human error, such as spreading the culture when the instrument was too hot, or not counting certain colonies separately because they grew into each other and into a large blob.
One thing we learned was just how ineffective antibiotics are when exposed to resistant bacteria. And as a next step, we repeat the experiment for a second and possibly third trial to determine if human error was the cause for the results not supporting prediction 3.